Hey guys, chapter 13 vocab video. First word is antigen or antigens. So an antigen is anything that the body makes antibodies against. Usually antigens are protein components. It's basically anything that the body recognizes as foreign and then makes antibodies against. So that's what it is. So bacteria have several different antigens all over their cells, viruses, etc. Even our own cell, even human cells have antigens on them, which is why when you get a donor organ, you have to make sure that they're a good match. In other words, you have to make sure that their antigens match the antigens in your body, or your body is going to produce antibodies against the donor tissue and attack it. So antigens are things that are attacked by antibodies. Epitope is just a specific part of the antigen. So if you think of the antigen as a big blob that overall is recognized as foreign by the body, the epitope is the specific site that a single antibody recognizes. So one antigen, there may be 100 different antibodies that recognize that antigen, but each antibody recognizes a specific part or epitope. Major histocompatibility complex, or MHC, these are markers or proteins on the surfaces of our cells that basically identify our cells as ours. So it's how your immune system knows which cells belong to you and your body and which cells don't. And so there's different classes of MHC markers that we'll talk about, but they're very important for identifying cells as self or not self. Immunoglobulins is a fancy mouthful, another word for antibody. So the sort of more medical word or biological word for antibody is immunoglobulin. They are synonymous, more or less. IG is the abbreviation for immunoglobulin. Clonal deletion. So when we make lymphocytes, B cells and T cells, they're very specific, and they go through a three-step process when they are being made. The first one is clonal deletion. This means that we delete any B cells or T cells that recognize our cells, because we don't want them attacking self. I don't want my immune system attacking my liver or my skin or my eyeballs, what have you. So if they recognize those cells as foreign, I need to delete them, stat. So that's clonal deletion. The first step is deleting defective immune cells, those that recognize self tissues. The next step is selecting for ones that recognize antigens. So if you have a bacteria invading you, there's a lot of antigens on that bacteria. And so you want to pick white blood cells or lymphocytes. You select for the ones that are actually effective at fighting off pathogens. And then you want to make lots and lots of copies of those effective ones. So clonal expansion is the process by which the clones that have been selected, the T cells and B cells that recognize antigen, then proliferate and make lots of copies of themselves, expand their population, and then go on to fight infection. So that's how the adaptive immune system works. We delete ones that don't recognize crap. We select for ones that recognize the bad things, and then we make lots and lots of copies of them. Aloe antigens. These are antigens that sort of come in a handful of different varieties. The classic example of an aloe antigen are the blood group antigens A, B, and O. So lots of people have A, lots of people have B, lots of people have O. So there's really only those three different antigens, but there are different versions of it, it's the same category. So that's the aloe part. Super antigens. These are things that are found on some bacteria produce these antigens that have the sort of ability to super activate the immune system. And as we've talked about, there's always collateral damage in an immune response. Just like in a war, civilians are inevitably injured. It's not on purpose as a side effect of battle. Sometimes civilians are caught in the crossfire. Same thing with the immune system. Sometimes your own cells are caught in the crossfire between a pathogen and the immune response. So certain bacteria or viruses can produce antigens that really cause a really big and bad immune response, and then you actually end up getting more damaged by your own immune response than the bacteria itself. So the classic one here is strep, which makes a super antigen that causes toxic shock syndrome. That shock element is actually shock from an overactive immune response. Antigen presentation cells, or antigen presenting cells, APCs, these are guys, they're phagocytes usually, and they eat bacteria or viruses, and then they present them on the surface of the cell and basically say, this is what you're looking for, telling all the other immune cells, look for this, this is the enemy, kind of like akin to maybe like taking an enemy, cutting their head off and putting it on a stake in front of the city. Or actually the best analogy is when you have like a search dog who's searching for a missing person, and you give them a piece of that person's clothing or something to sniff for, so now the dog knows what it's sniffing for. So these antigen presenting cells basically present little pieces of the pathogen to the other immune cells so they know what to hunt for. Cell-mediated immunity is a term that we use for the type of immune reaction that T cells do, where they kill cells. It's cytotoxic, it's deadly to infected cells, versus humoral immunity, which refers to antibody production by B cells, also known as immunoglobulin production. So B cells produce immunoglobulins, and there's different classes of immunoglobulins. IgG, it looks like a Y-shaped, so antibodies or immunoglobulins are Y-shaped. IgG, they're just single antibodies, and these are sort of the mature antibodies that are responsible for fighting off infection, that also can stay circulating in your blood for long periods of time and help provide some immunity to future infections. IgM is the one that's made before IgG. So when you first get an infection, the first antibody that you start producing is IgM. And IgM is pentavalent, meaning it's 5 of those Ys connected together in like a snowflake shape. And these are sort of the practice antibodies. And if they work well, then the B cells mature and they start pumping out IgG. So IgG is like the gold standard antibody. IgM is like the practice round. IgA is divalent, meaning it's 2 of those Ys connected together end to end. And again, just a class of immunoglobulins. These are found in secretions, so in breast milk, mucus, snot, very heavily found in secretion, so it's sort of the secreted antibody. IgE, I don't have a picture of it. It also looks a lot like IgG, and it is the antibody that's most associated with allergies, so it's the allergy antibody. I don't have a good way to associate E and allergy. Think allergy, IgE, allergy, I don't know. Colostrum is the breast milk that's first produced when a breastfeeding woman, the first milk that she produces, it's thick and yellow and only lasts a couple of days, but it's super nutrient dense, but it also has a lot of antibodies. IgA in particular, because it's the secreted one. And it helps to protect a newborn from infections. Vaccination is also known as immunization. It is injecting someone, or sometimes there are some vaccines that are ingested, you eat, but most of them are injected, and it contains either dead or inactivated bacteria or viruses, and it basically just exposes you to a small non-disease causing dose of a pathogen and allows your immune system to elicit a response, its own natural response that it would have if you actually were exposed to that pathogen. It's exposing someone to a controlled amount of a pathogen and then allowing their immune system to build up immunity. So it's really a powerful protective medicine that we have, some of the most powerful medicine that we've really ever created, because it's one of the few medicines that can prevent disease, which is pretty impressive. And then adjuvants. So an adjuvant is a component in a vaccine that helps to stimulate the immune system. So if you just injected a small amount of bacteria or virus in somebody's arm, the local immune cells might clear it, and it might never get, to that third line of defense, stimulating that third line of defense and really getting that memory response. So I like to think of an adjuvant as like something that tickles the immune system, or maybe even really irritates the immune system. Sometimes it's a substance that just holds that bacteria in that spot for longer and makes it last. It's preservatives can serve as adjuvants. So it's something that really amplifies that immune reaction so that you get a good response to this very small amount of antigen in order to get a good, strong immune memory to it. So, the end.